Guten Abend und wie geht's? Wie Leute heute geht's ab? I just said a very good evening. How is my all families doing? How's my students doing? What's up? What's geht up? It's in German. Um, very good evening. Very good evening. Good evening, everyone. Good evening, students from all over India. I'm looking forward to today also, like yesterday. Thank you for your great response. I got a lot of messages from you late night. Um, I want to sleep by 10, but yeah, it took me some time. Uh, yeah, uh, I'm also good today. Uh, I had some work today in the morning. I was just preparing for your slides and taking care that we do not have lagging today again. So yeah, I removed my video coming up there because of that might be we were seeing that fact. So so to keep the streaming going good, I tried to keep up the best resolution for us. This is technical IT stuff, and yeah. So how was your day? How was your day, students, today? How was your day today? Today was Wednesday, right? So what did you did today actually? Yeah, I will try. I will speak louder as possible. Um, good, good. It was sunny or rainy at your side, as monsoon is happening. Yeah, online classes everywhere. That's the future. That's the future. Online classes. Um, all Zoom, Google Meets, WebEx, YouTube streaming. They are the future now. They will take over our online classes. Though, to me personally, COVID 19, this pandemic, is to some extent in Delhi. I'm from Delhi actually. I'm living in Delhi at the moment. So when I go to some Crole Bagh market or some other market for my personal shopping, I never think of that there is any COVID-19 is happening. Everyone is living such a normal life here, and I don't know how many of them are being COVID positive now. So uh, yeah, today I will be dealing with your two topics. That is, yeah, a good question, Bhupinder. I remember you are from the pharmaceutical sciences in the Punjab University. I mean, uh, me personally, I like more offline teaching where I could interact with my professor face to face. And I have my student members also with me. So we can discuss just when the lecture is over, or we have any questions, we can directly go to the professor. I could ask there. Online is somehow have some barriers, some cons are there, but on other side, like at the moment we are able to go for this class for 400 students, almost 400 students there, and we also have yeah, we're sharing the workshop, virtual workshop, and yeah, we are doing something which is not possible, which is possible, okay, with the live, but we are doing something being having a virtual workshop. and making it happening today. So that is something we are doing, something impossible. Uh, yeah, no one, please, please, please. I repeated in my last class also that please do take care of two things. Carry your sanitizer with you, put it in your cycle or your scooter or in your car. Always keep it, but don't use it too often. It has also some side effects. Using of too much of sanitizer, it might be allergic to someone after some time. And keep washing your hands with soap. That's it. A normal soap, any soap or Dettol, anything. Washing hands is much better than sanitizing, actually. Um, yeah. School, I wish they could open just now, but it doesn't make sense at the moment because most likely you childrens will be really vulnerable to if somebody one is sick of COVID, it could spread to masses. Even though all the big IT companies, big everything is going on now, nobody much care about it. All the malls are open, everyone is shopping like it's all normal again. So school should also be open the same way. And also I'm seeing this NEET exam and JE exam dates that students willing to not to give. Actually, they are right. Why they should like forsaken their life while going for the COVID-19 now? So that's makes a lot of sense. So who people they are missing their labs, I will recommend to start something with the bioinformatics stuff by staying at home. So I will share a little bit of bioinformatics techniques here to not today but later. So you can try here and there some computational biology uh, to see or to see just check COVID-19, how the genomics of COVID-19 is being made. So all people are doing research, how things are happening. So we will also today put some thoughts on the your your internship projects and your assignments. Yeah, so both things will be dealt today also. So it's at the moment 19 for 50 and almost 95 members are there. Let more members to be added more here, then we can start in 10 minutes or so. Then I will explain something. I mean, I stayed in Germany. So what they do is for conducting examinations, they from till seventh standard or sixth standard, no, not sixth standard, but till 7 or 6 years old, they don't let these children to go to school. The school starts quite later, quite late age, and at one point they transform the students into three parts, one in gymnasium, one in realschule, and one in some even lower standard. So the one who are in the lower standard, they are not so good in studies, so they are told that you have to work the laborious work, but the laborious work is quite equivalent paid as you're working being educated. Then comes your Realschule, where middle IQ students are there, so they also could think of doing an white collar job. And gymnasium people are extra intelligent students, so they are the yeah, future scientists, future doctors, or something. So when they're done with the schooling, according to their level of schooling, they had done gymnasium, Realschule, or other one, the first one, they are transformed into that part. And for medical, they have to give an entrance, but not like national entrance level like in India we have. In Germany, it's different. It's actually state level, and it's a public university. And best part of Germany is that you don't have to pay any fees after you are done with your schooling. Even in schools you don't have to pay, it's all government funded. Your whole bachelor's, your master's, your PhD, everything is government funded. So government really take care that the education the students they are taking, it should be free, and there is very less privatization there also. So, Bache, many people ask me about PhD. For PhD, Uh, After your masters, if you have done your masters from India, look for your professors who did in your what you did in your master thesis. Then whatever the work you have done, just check whether the publications of your professor, they are matching to your work. Only then a professor can seek for you to have a PhD for 3 or 4 years with you, because in Germany it's 3 years of PhD and you can finish it within given time, and you can also have publication out of it. So it has lot of advantages, I have to say. So no money for PhD in Germany. It's all free. You get paid actually. I was paid around 1600 euros. Just multiply by 80 and then you can find it out. So I was getting around more than 1 lakh per month for 3 years continuously every month actually. So I saved a lot of money also from there, which still I have. And yeah, so it's a good investment to do your education in Germany. I have to admit. Yeah, France is also a very good options. It was not opening before because most of the French people, they don't like to speak English for some strange reason. But now they are opening students for international students also. They are trying to speak English, bringing English in their curriculum, like Germany did. So in Germany, everyone speaks English. Not mainly the old people, but mostly young people. They are speaking in English, so that's fine. That's fine there. Yeah. Yeah, like in India, we have this government and private universities. We have lot of huge amount of private universities that the normal student couldn't get the admission. Only the high IQ students, or and we have a lot of population also believe in that. And whereas in Germany, it's quite less. So the chances of getting to public university in Germany is quite easy. It's not so, you don't have to fight so much with so many students. So everyone gets what the university they want, what they want to study, what they want to do a career. So it's all open for everyone. So here, just because of our big population, less opportunities to get admit in the best universities, is somehow playing a kind of role there. So mainly for masters, if you're going for masters, mainly TOEFL or IELTS is enough. You don't have to give GRE. But if you're going for some very mechanical engineering course where at high-end university like Technische München University or in Hamburg University or Frankfurt University, then GRE might be important. No, no, SAT is not for masters in Germany. Mainly IELTS is enough. Yeah, I will. It's 55 now. I will. It's 153 members we have joined so far. Let the people add. Then we can start with the training and in project assignments things how we have to go for it. Yeah. Sometimes I'm saying yeah. It's just it's came from Germany because yeah means yes actually, and it's written as JA, but it's spelled as yacht like Y. So it took me quite time there to be there like that. That's why. Yo, so yeah, IELTS academic, yes, of course. General is for migration. You have to give IELTS academics. Just get 7 bands. 7 bands are enough. It's more than enough for you. Every day, notes will be provided to you. Don't worry. That's the beauty of this workshop. Each day, whatever we are studying here, we will provide you. And yeah, just keep giving. Be disciplined, respect each other here. Um, Whatever the level of teaching we are doing, we are giving you the utmost best level here. And don't think like, oh, this is something that or fabricated. No, it's not. We had some students from our previous workshop also, and it's meant just for students, not for us. And each presentation has been made for you students, so that whenever you left this workshop, you can remember them, that you did all these things. Yeah. So I have my at the moment a book called Principles and Techniques of Biochemistry and Molecular Biology by Keith Wilson and John Walker. Wilson and Walker is the most famous books for your principal techniques in biochemistry and molecular biology. And I had created, if you go through the book, I can also provide you book also on the telegram later. But the 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 index system of the book says that first days is the introduction, then it says cell technology and cellular basic cellular cell culture techniques and microscopy. So I try to maintain the same order as per the book says, to my best level, and then later on I modify a little bit because it was too much in the book. So it's a mixture of my own personal techniques that I have that I had learned, and I can bring it to you in during this workshop, and also with the help of the book. Like I also took some presentations from Gene's 11 book, um, and also some from biochemistry, Lahinger and Harper, which is for medicinal biochemistry. So I took quite a range of books and facts to make these presentations. So it take a lot of time for us to do so, to give you the best, uh, and to get out best out of you also. Yeah, Harshil, I found your message that you want to do something with the bioinformatics uh, and NGS, next generation sequencing. You're all open to the things that you like. I will just explain. Let more people add. So it's been 192 so far, and it's almost 8 o'clock. Yes, it is. It is 8 o'clock. Uh, yeah, yeah. Gary Walsh is also a good one. These books just absorb them, because the foreign authors books, the way they had created with different cycles and how they explain everything in so detailed manner, it's like a fairy tale. Yeah, if you start one chapter, you want to keep reading it and want to finish it until it is done. So read these books as a story books, actually. When like once you have this Harry Potter book, if you once start one series, you can't stop to see what will happen next, what will happen next. If you're done with the TCS cycle, okay, what's with the Kelvin cycle then? How is that working? um, so, yeah, CRISPR labs. Yeah, it is happening. It is happening. CRISPR is hot. It was happening in during my PhD time when I was doing my in my 2016 and 15. CRISPR becomes hot, and it's everyone is doing it. Everyone is doing the CRISPR thingy. So, yeah, I can take small session for PhD, of course. Uh, so today is the day for cell culture and microscopy. But before we start, the person who are doing the internship with us, so what I have decided, so I shared already in the group this topics for the internship. Yeah. So, gene therapy, RNA interface, genome editing, drug delivery, DNA synthesis and sequence, stem cell research, maturation of sperm cells, genetic engineering, monoclonal antibodies, cancer cell research, primary cells and transfected cell lines, cytokines, growth factors, and cellular processes with growth to achieve the appropriate size, predictive models for cancer cell specific dependency. So, I just wrote whatever I felt these could be the topics. Like I randomly choose these topics for you. But if you want to do something else beyond these topics, you are most welcome. Okay, you are most welcome to do so. And what you have to do in this project is, like, let's say I show you my publication. We published in 2018 from Germany, three universities. Um, SR. So it's in Nature. In publication, it's quite yeah. So it's impact fact. There's a called impact. Sorry, impact factor, which means how much does it has impact your publication. So this publication has around at that time 4.5 to 4.5 to 50.0, but now I don't know how much does it impact factor in the moment. The scientific reports, it's under the head of Nature. So as you can see, I have my friends Simone Lemely, Haiso Gize, Suzanne Kamer, Stephanie Meyer, and the most of the thankful person I am during this publication is of both my professors Ali Al Moush and Florian Dichter. So they both really helped me to cross this huge Mount Everest. But, getting a PhD is like a Mount Everest. You have to do a lot of hurdles. There are a lot of things are happening with you. You are sometimes homesick, and you sometimes your experiments not working. Sometimes. Yeah, there are trillions, zillion, billions of problems, but you still came across all of them, and publish successfully, defend your PhD successfully, and done with your PhD. So if you have decided that in this life, before you die, I want to have a doctorate degree. This was my dream also. So when I was in my 12th class, I was not able to clear PMT exam actually. So I took option of BTech biotechnology. But I have affirmed myself that before I die, I want to have a doctorate PhD name in my front of my name. Whether I'm not a medical doctor, but a molecular medicine is also fine. Is okay with me to live. I can live with that. I can live with that. So to continue with the topic about your transition, about your internship. So this was the paper. So here you can see author's name. So first of all, there is a topic, the name, the title of the topic. So keep that in mind when you are making your project. Have a very crispy and small and succinct title there. Yeah. Then and the members, how many members they could make these assignments. It's sorry, the project. It's up to you. If you're living all together 5 members, go for it. If you're 10 together, go for it. Do it in the group. Just make small groups or large groups up to 10, and try to and write the names like this. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So 11 members are there. And also, I will share this paper with you also later. And please try to write like who did what. So what are these people did while making the project. So like let's say this is author contributions. So who designed the diagrams, who make the figures, who write the abstract, who wrote the review, who got the later literature reading. So like this, you have to create this author contributions. So in this, we did like experiments and so like Florian Richter and Ali Al-Moush did design the study. Then Susanna Kramer, Susanna Lutz, all these people did the experiments. Simranjit Singh and Simone Lemely, and then further these people perform the experiments, and then further they analyze this data. Then they give the technical support and advice, and these they wrote the whole manuscript. So like this way you can rewrite the names of the people who write the manuscript, who write the review. So you have to write a review actually. So in review of these topics that I have shared with you, uh, please write down the abstract first. Please write down in your notebook. There should be first is your abstract here. Yeah. So like a small summary of the whole thing, how it has to be done. Then comes your introduction. A little bit of introduction starts. Then all the just do some literature studies like I did in this paper to write about the ROS. So my work was related to the oxidation of proteins in the in the cardiomyocytes. So how oxidation of proteins could modify the disulfide bridges and leads to the change in the modification of the protein structure at the post translational level, at the PTM level. So I will explain about my work later, but just I'm giving you a little gist at the moment how you have to write your review. Then uh, just get, so I don't want big reviews from you. I don't expect a much from you. So I want at least 3 pages or 4 pages of your review to be there, with all the sections of with abstract title, people who have contributed, all the things that you can add into that, like if there's a method you want to think of, then whatever the like succinate, you can write about the various years of how the research of that method has been produced. Or if you're looking for some technique or some gene therapy you want to explain, then just explain like how gene expery came to the world and how things were working with it. So you have to write a summary so that when reading, somebody is reading about that topic, they understand it and they can make a sense that you are bringing this topic to the world. Yeah. So then at the end, so you cannot do experiments here at the moment, sorry for that. So where you in a research paper you have to write introduction, then comes this is quite opposite actually. So here we have results also like this, and each figures have to be maintained with figure legends. So keep remember, when you are adding figures you should have a figure legends also, with A B C D, and then methods, like in this case we have added methods also. So then at the end is discussion, and that's it. Then at the end they have produced this material method. So every journal, so this was a scientific report, so they bring the material method at the end, but most likely it comes after the introduction, then comes the results, then comes the discussion, then comes the conclusion. So each paper has its own way of it. Then at the end is the references. Keep take care that you please also put references like this, not the same way, but the best you can write it manually, or you can use software like EndNote to write these references. So this will be quite huge task for you. I'm pretty sure for the students there, this will be quite task for you to do so. You can even publish if you want whatever work you have done. You can ask for free journals to publish your journals actually to your publication. I can also help in doing so. Just keep sending me, make a group, just tell us your topic, what you want to do, and we keep continuing with that. So I will not. I take have taken 10 minutes so far. And about the other thing, internship is done about the assignment. It is not compulsory that all should submit, but if you can submit, that will be really good for you. That you will. You had accomplished something. I got really good assignments today from many students, in my email, in my telegram. So it was, so maybe this was Mohammed Anjum. Uh, so he sent me his assignment today with such a, you can see it. I can see some work has been done, some effort has been done there. So they made this nice diagram where your things were happening. A small summary about cell culture, cell, yeah. uh, they made by themselves these graph, I guess. So basics of molecular biology, then some summary about that. So this is, I'm just explaining within 2 page. Okay, it's a bit 3 page, it's fine, but still I really like it. So everything within 2 pages, very nicely done. And I told you that you have to send your assignments at the end of the workshop. But if you're also sending me every day, that's also fine with me. But if you feel like to combine them all together, that's and bring me to the at the end of the thing, that's also fine with us. So I think your all queries, your all things are clear now. No doubt so far. I will start with the workshop of today, and I have to be slow as I was told. So let's just start with the basics. Microscope basics. Then we will go to the advanced version later. So I'm pretty sure you all had used microscope in your class 10th or class 12th standards to check mitosis or meiosis in onion bulbs or in some other watching some maybe yogurt curd under the microscope and seeing billions zillions of bacteria moving here and there. So this that was the fun part, but this technique was very limited. We have taught in the school level, but now it has been ranged to very high level using electron microscope, scanning electron microscope, phase contrast microscope, dark electron microscope, um, inverted microscope. There are a lot of the closed incubators microscope. There are so many microscopes are available at the moment in the market, which can help to do whatever the applications that you're looking for to look for that. So microscope. So what does it does? It magnifies things to make things looks bigger and helps the scientists to live the things to see the small things with naked eyes. So like see here you the crystal of ice, how beautifully we can see them, uh, the red blood cells, all these things. So this is the basic of your basic microscope, the structure. Here is on the top is your ocular lens, then comes the arm, then there's a stage, then there's a coarse adjustment. Like from here you can make magnify up and down, fine adjustment there, base is there, then from the bottom light is coming up if you need it, and there's a diaphragm to open it or close. Stage clips are there, objective lens 10x, 4x. Remember, yeah, 10x, 4x, 20x lenses. Then there's a nose piece. This is called nose piece, and that's the whole microscope looks like. And you always have to carry it one arm and base with other hands when you're moving it. So there's a simple microscope who has only one lens, and then there is a compound microscope has two set of lenses. Okay, so it can magnify up to 100 to 200 times than the really are. And the third one is electron microscope, which could magnify to 300,000 times, which the normal lens is not possible, and we can see things at the electron level to check that. We can see things with the help of by using of electrons, by initiating the electrons into our cell, and we can see our figures then. Oops. So these are we have discussed this. So on the top is your ocular lens, then the coarse adjustment here, then the fine adjustment here. With the you can look from this part. From here you make it knob up and down into focus, and then from here is to do the fine adjustment, like to see it much better even than before. Then comes your arms. You can see then the body tube here at the bottom, and the nose piece. Yeah, so this one is your arm here on the side, this is the body tube, and then it's on the nose piece this side. Higher power objective lens, so usually is the highest is the 40x, low power is mainly the 3x to 4x. Yeah, and middle power is around 10x to 20x x like that. So all these different lens power are fused into the same part. This is for someone who have never used microscope. That's why I just try to have a basic about. If you know about this, then we are good to go for for further slides. Then there's a stage, clip, diaphragm. So this one is the clip, stage, and then at the bottom is a diaphragm that controls the how much light you need to be see your stuff, that specimens that you're looking for. Clips are meant to hold your slide in it there. Okay. So a light source from the bottom, yeah, and the base, which helps to rest of the microscope. There is base. So this is all clear. Very einfach. Einfach means really in German means it's quite easy to understand, quite easy to go for it. So let's start. We have done the basics now. Can you tell me what will be the what thing from the right if we start? Yeah, please type. I will go forward then. So this one is first is your ocular lens. Then this is your body tube. Then the nose piece. Then this is your objective. Then on the left is stage clips. Then what is this thing? Diaphragm. Then light source. Then the base will come. Sorry, arm on the top. Then the stage will come. Then a coarse adjustment, then fine adjustment, and then the base. So this is your simple microscope. So how things looks in the simple microscope is actually looks opposite. The field of view in the area, whatever you're seeing there, is through the eyepiece actually. So if the E looks like we write E like this, so E would be like opposite and on the other direction. So whatever the things that you're seeing is yeah, upside down and inverted. So if the 7x is this, comparing the magnification, your 10x would be like this. Yeah. So we can see better details with the magnifications which cannot be seen with such image. So like if we just want to see this kind of magnification, we can enlarge this to this much. Okay. Then how you can check the power where it is written. So here, so this is like 10x 10, this is 40. So these are the various power of your objective lens is written. So how to calculate the magnification? You just multiply the power of eyepiece multiply by objective. You multiply these both objective lens, then you can calculate the magnification of your microscope. So to find the power of the lens, it is found on the side of the lens. Magnification power of lens is always identify the label x like 10x or 1000x, and then you multiply with the eyepiece power. So this is an example done for you. So 10x times multiply by 100x, so magnification will be 1000. 10x times 50x, the magnification will be 500. So 10x times 40x will be 400. Yeah. So as I told, it's upside down and backward what you see. If E is like this, you see like that. So E goes like this and this and this. So it's different. So we don't have microscope at the moment. Sorry for that. Otherwise, we have done this workshop and check everything and record some observations. Maybe some fruit flies, bring them under a microscope with our tiny forceps, and then check how things are looking in there. And but this thing is quite interesting, how to make wet mount slide whatever the specimens that you're looking. So this will be nice. Use a dropper. So to do so, use a dropper to place a drop of water on the center of clean side. Okay, then with the help of tweezers, lay your specimens on the drop of water, then gently touch the cover slip. You can see like on a 45 degrees Celsius on the edge, and cover the water and then slowly put it there so that the no air bubbles are there. That's the main thing to do so. So like this, you have to cover slip, lower slowly. Okay, so this is the scientist who is checking genes actually. So this gene has three pairs of legs. So it says it's a mutant genes, and then the microscope is saw. It's a funny part. Okay, thank you very much for this. So it was just short introduction about microscope, so that you know how things are, how things looks like. So next is I have a lot of things to do to deal with you today. So please be with me. This is around 42 slides, so it's a long one. Okay, so in this, actually, I will not only deal with light and electron microscope, but its various types also. How various microscopes, various microscopy is beneficial for us. How we can see different cell lines or different things at tissue level, or we can mount the cells on the slides permanently forever. Um so that we can have the proof of our results for quite number of years, quite number of years, yeah. So this one, first one, that what you're seeing is a microscope constructed by the Leeuwenhoek in 1670s. It's present at the moment in the medical library, University Texas Medical Branch, Galveston, Texas. So this was his way of making the first microscope by Leeuwenhoek. And this one you can see is a replica of Kulper tripod microscope built in 1725 by Edmund Culpeper. Sorry, it's again in the antique part in there. So you see how nicely the wooden touch they have given it, and with the golden frames outside, having a eyepiece, objective lens, and then the mirror at the bottom, and then the uh, a stage there being set there. So that's some past old school microscopes. This is a replica of Marshall microscopes around 1700 by John Marshall. Uh, this is also quite, yeah, looking quite antique. It would be nice to have something like that on our, uh, in front of uh, our study table sometimes, to just to check things, how things are. So we talk some other stuff. Let's come to the point. So the content of the this workshop would be dealing with the types of microscopes, yeah, and specimens preparation. Okay, how different types of microscopes are. Within the types of microscopes, there will be two microscopes we will be dealing: light microscope and electron microscope. Within the light, we will deal with around seven microscopes ranging from bright field to confocal, and in electron we deal with the SEM and TEM, scanning and transmission electron microscopes. And on the other side, we will see how we can make specimen preparation, like we just checked in the previous slide, how the microscope was made in that side. And we can check scanning electron microscope and transmission electron microscope, for them how we prepare the samples also. Okay. So yeah, microscopes have been very essential for the tools for the cell biologist. If what we see in front of our eyes, whatever the things that we are seeing, is makes sense at the end. If things are not happening in front of our eyes, we cannot say that this is happening or not. So this is a direct proof of any mechanism, of any change, of any alteration within the cell, movement of the cell, or engulfing of the products, or production of proteins. If we can see all these by our with the help of microscope, we can say yes, this is happening, and we can be quite sure of that thing. So mainly scientists are divided into two broad categories, that is light microscope and light microscope. And this one is the old, very old molecular bright field microscope, fixed stage with the mirror. So you can see some very old microscopes. So starts with the first light microscopy. So this is a bright field microscope, the normal one that we have discussed before. You can see this is the stage is quite strongly made, not like the before what we saw, and it has two eyepiece to check the things, and it has one, two, three, uh, four objective lens, yeah, to see your slides with different magnifications. It's a bright field. It's called as because of the name. It's a bright field. Field will be bright. There will be bulb at the bottom which will view the dark side, and we can see our slides in very nice manner. So this is the most common general use of microscopes. Bright field microscopes are named because the microscopic field is bright while the objective being viewed is dark. So it's a simple design. Light directed at specimen is absorbed at the form image. Unstained specimens have poor contrast. It's quite ideal for the bacterial cells and tissues. Uh, it has a quite bright background. Dark specimens are possible. And a halogen light source we are using in this case. So this is what it looks like in a bright field image of our blood cells in a peripheral blood smear. So you can see your blood cells stained here in this part, and this is a section of a gut tissue where you can see ciliated parasites. Ciliated parasites are being seen there, and this is a flagellate trichomonas we seen in the bright field microscope. So we will deal like this now. We will check each part topic, like each microscope we will see what are the types, how are the function is happening, then we see one image afterwards. We go like this now. um, so here you can see also an inverted bright field microscope where your light will be put from the top instead of the bottom, and you can check this is the eyepiece will be here to see your things. This is what I use actually for my PhD to create some videos. So to set that, I will show you very nice heart pumping videos now to you. um, yes, this one. So what I did in this experiment, I have cardiomyocytes. Cardiomyocytes are the cells from our heart. So what I did from the neonatal rats, I took the heart, and then from there I extracted the isolate neonatal rat cardiomyocytes, and then from them I made four conditions in a cell plate that one is control, one is 100 micrometer, one is 1000 micrometer of peroxide. So I check in different concentration of peroxide what happened to the uh, cardiomyocytes. Okay, this is not opening. So this is in control condition cardiomyocytes. So here you can see this flagellated body. In the center is the nucleus, and these are air bubbles. Just don't worry about them. These are dead cells actually on the top. So you can see how cells are movement. So this is actually heart is pumping in this case. So I will show it again. So this was done in the inverted microscope, this image what I'm showing you. Now what will happen at 100 micromolar of H2O2? This oopsie. So this one is 100 micromolar. So this movement is slow now. It's not stopped. Cells are not dead yet, but movement is slow. So this is around. It took me almost 24 hours to create this video, only for 4 seconds. So yeah. Now let's see at 1000 micromolar. What happened there? It's just stopped. The functioning of the cell just stopped at 1000, and at 10,000, which is quite cohesive for the cells, you can see that cells start to coagulate and destroy themselves, and they suddenly started to pop out. You can see these bubbles coming up suddenly. Yeah, that's the thing. That's what happened when you put a lot of peroxide on your cells. The cells will, yeah, cells will die. So you can see in these four different conditions, just check their images, how a different concentration of your reagent could lead to the effect on the cells, and this done all with the help of microscope. If microscope has been not there, so all these research thingy was not possible then. Okay. Now, next is your DF, dark field microscope, which creates a dark background. So it creates very dark background first of all, and then you add DAPI. DAPI is like it's a dye which we get it from the fluorescent jellyfishes in the bottom of jellyfishes, yeah, not from the jellyfishes. We get this GFP, green fluorescent protein, GFP, and we insert into our cell, and we create a dark specimen in the background, and then we check our objective lens in high contrast with good like this one. So you can see it's a dark background in behind, and here we can see an algae, hydrodicton reticulatum, with the help of dark field microscope in this side. And on the right hand side is the leptospira, which is viewed in the dark field microscope. We have a dark field in background, and we can see our microscope like this. So this is how it's shown. Then comes our phase contrast microscopes, which converts differences in the refractive index. So mainly the refractive index here, the specimens we can see differences in that image with the help of brightness. So central portion of the light is blocked. So whatever the source of the light is blocked. By creating a ring of light from the condenser, we create a ring that illuminates a specimen. So like from here, angular ring. The light waves refracted by specimens were slowed by phase retardations, increasing the difference in the wavelength between the refracted and the unrefracted rays. So they increase the difference between the wavelength of the refraction and unrefracted one, which do not pass through the phase plate. So this is technology is from the 1940, provides quite high contrast, good resolution, good for bacteria, flagella, cilia, organelles like mitochondria, good for unstained. If we have not stained something, we can see things live mounted on our slides. So like this. You can see on the left side is the unstained squamous epithelial cells observed with the phase contrast microscope. And the right side is the DIC microscope. We will study about this also, and we can change the main difference is here with the help of refractive index. We can check at the different level of our image with the help of this. Then comes our polarization microscope, which is good for bright image, dark background. You can check crystals of minerals, molecular structures, and we can also do quantitative study in the polarization microscope. So it detects the specimens that are bifringent. What is bifringent? Which has double refraction. Velocity of light refracted substance is not the same in all directions. So whatever the velocity of light is being refracted, it will be not in all direction, but but on the double refracted it will be. Specimen is between the two polarized crossed at 90 degrees Celsius to each other. So one condenser, one is objective. So here we have a condenser here also, and we also have a polarizer in this thing. So this is how the image looks like. This is a Martian meteorite from the Mars on the left hand side. Meteorite was came to the Earth, and we check it with the help of polarizing microscope. And on the top right side is a bright field view of the renal epithelial cells containing the fat tubules, and the bottom right is the high magnification view of same cells viewed with the polarizing microscope. So you can see there's so much difference between this and this. So mainly polarization microscope is done to check crystals and minerals display, to check the patterns in this polarizing light. How the minerals looks, the patterns are as per the Mars rock and also our Indian or Earth rock. Then there is epifluorescence microscopy, which is done also with the help of UV light source and mercury and xenon arc lamp. So on the from behind we have UV light also coming up there, and we have this barrier filters with the diachronic prism inside it. So this is some advanced version of it, bachcho, that you can check advanced level of your studies. So this is what you see in the epifluorescent microscope, is the tetra tetrahymena. It's a ciliated protozoan with the fluorescent dye, that is fluorescein isothiocyanate, which emits green light. So in this we use the fluorescein isothiocyanate, which is emitting a green light in this above thing. And above right is the cell is stained with the antibody against cell membrane surface antigens. So against the cell body we are seeing things with the help of antigens here. And on the right side is the bright field view of the tetrahymena. The same thing, that it's look like this and it's look like this. So in bright field it's quite hard to say many things, how the pattern looks like, how the cell membranes looks like, how the pattern looks like or outside. But with the help of dye we can see, okay, this patterns look like in this very straight striated manner, and it's in an order, and there is a like a shunk, like these things inside this is happening. Now students, this is also one of the image from the epifluorescent where we can see seeing the metaphase state. Remember metaphase, your all nucleus, your all chromosomes are separating above from each other, and the center, your centrioles, the spindle fibers are getting separated. So very nicely shown in this cell figure. So they have dyed the cell with a different dye, and they made the centrioles dye with a different dye, and these chromosomes being separated with a different dye. So the three different dyes has been added, and then this image has been made. So you can see the red is keratin filaments, yellow are spindle apparatus, whereas uh, tubulins, where blue is your metaphase chromosomes. Okay, so we discussed previously about DIS, differential interference contrast microscopy, also called as Normansky optics, because it might be found by the Normansky person. So here we have, oops, sorry, let me put this there. So here we can see in this microscope resembles phase contrast, but more sensitive, higher resolutions are there. It's give 3D images. That's the best part. It's not earlier. It was always we are giving 2D images in all most of the microscope, but in this we can see 3D images and excellent resolution in them. And unstained specimens, live mounts can be seen within the cells, and we can detect refractive index of specimens in it. So this is one of the DIC image. You can see it's a cricket or ovarial showing oocytes. So we are seeing the oocytes there with this O, and Balbiani bodies. These are Balbiani bodies like on this side, and this is whole is your oocytes. Nuclei are the oocytes nucleus with the N, and this is also called as mitochondrial clouds. These ones, Balbiani bodies, and yeah, this bottom you can see the 3D structure of it, how Balboni body is moving from here to there, and it's stained with the rhodamine 123, specific for the mitochondria. You can see a 3D structure also in it. Now we are done with the various microscopy already in our light microscope. This is, I think, the last one, confocal. Yes. So this is quite advanced technology, and most of the researchers, they whenever they are doing for want to look for a broader range, like to check things in more advanced version. So they go for this confocal laser scanning. Even for my PhD, I was doing bright field microscopy mainly with inverted microscope, but I got chance to do the confocal microscopy also, but I was not allowed to do a lot of time there because it's quite expensive, and teachers are not like willing to spend a lot of money research to students research in that way. So this happens sometimes. You have to uh, maintain finance also to keep experiments going. So just a second. So in this case, a laser is focused at a plane in the specimen and scans the specimen in the horizontal section. So in the horizontal section, we check how things are, and only light from the plane of focus reaches a detector. Then the scan image is digitally recorded in the computer with the nearby, and then we have a 3D image which can be seen with the help of this confocal microscope. So it has argon laser. So we have argon laser working in this case. We have high resolution sharp images, high sensitivity can be used in the fluorescence mode and also reflectance mode. Beyond that, you can use it for the we can eliminate the background interference in that case also. This is quite old microscope. You can see even the PC is very old. At the moment, it's very advanced versions are there, which has even the hydraulics at the bottom to maintain the your microscope going good. So these are the various images you can see from the confocal microscope. On the left is a trichomonas immunostained with antitubulin and fluorescein isothianate. FITS, we call it FITS. So green structures consist of the microtubules. These are the microtubules that you're seeing. Okay. So this is the posterior flagellum, and AF is the anterior flagellum here. C is your costa, called as costa, and exostyle, and the red arrow is the exostyle here. And this image is the bright field image of the same thing with posterior flagellum. You can see how the same image that it looks like here, bright field in confocal, how advanced version it goes like then. Now, some bigger microscopes. Would you like to have some 5 minutes break now? So we can start from here only, electron microscope again. Just take a 5 minutes break. Then I see you soon then. So all is fine. So I will just come within 5 minutes. That's 45. So hello class. I was for break, बच्चों. I'm audible now. Okay. Thank you for giving me some time. I recovered myself, and you also might have recovered. Um, because I'm telling you this technique in such detailed manner because this will be really important in your future wherever you go. If you know that there are these much of microscopes are happening, so you can ask your PI means principal investigator, that I want to use this microscope for this application. So this will be really helpful for you then in that case. So now scanning microscope and transmission electron microscope. Thank you. Um, so in the scanning one, we mount our specimen surface coated with the gold, platinum, or rhodium. Yeah, so we use gold or palladium or rhodium as a surface coating for to check at the scanning electron microscope. Then specimen is placed in the vacuum and an electron beam scanned back and forth over it. The electrons that bounces off the metal coated specimen surface are collected. Okay. Then after the we have done the coating, then a beam is been bounce is been put on up our thingy of our specimen. The beam that has been reflected back is collected and converted to a digital image and displayed on a TV like a monitor. So this is the main principle of your scanning electron microscope. So electron beam is focused using a magnetic field. So electron beams that we are sending it, we maintain it using a magnetic field. In that case, it's provide a 3D image, gives information about external topography of specimens, and much higher resolution and magnification than possible in the light microscope. So here you can see a comparison of SEM. Uh, of so both DIC and SCM reveal specimen surface topography, but much greater details are using SCM because of greater resolution and magnification capability. So on the top left is a scanning electron microscope on here, this one of a trichomonas, and on the right top is a tetrahemna. So you can see so how detailed you can see. It's like a seed with a corpse coming out of it. This is how your these microorganisms looks like. And bottom are the DIC images of the tetrachomonas and tetrahymnus. So you can see how it magnified your image, how magnified your structure looks like, how you can see your organisms, microorganisms in such a detailed manner from this level to this level. Next is your TEM. In this, we have dehydrated specimens. We remove all the water embedded in the resins, harden and section and stain with heavy metals. And then, with the help of uranium or lead, we insert them into the electron column in the microscope. So electron beam is absorbed, deflected by the heavy metal stains and showed and cast on the film or the phosphorescent plate at the bottom of column. Instead, in the previous we had a metal coating outside, but in this case, it is being absorbed and then we see the a 2D image instead of 3D image. We see a 2D image in this, and we can see the internal cell structures in this TM, and high resolution, high magnification is possible. Electron beam is also focused by the magnetic field. So on the left is your SCM. You can see we can see a 3D structure, that's good, but on the right we can see the cell structure, cell organelles inside of it in such detailed manner. That's the beauty. So we can see G with the Golgi here, H with the hydrogenism, F for flagellum, yeah, RF is a recumbent flagellum, and this is a cross section of a interior flagella. Your whole flagella looks like this with the help of your TM image. Some more. So here you have rough endoplasmic reticulum. Um, you have your nucellus here, nucleolus. Sorry, nucleolus. This is your nucleus. This is your mitochondria here. This is your cell boundaries. You can see very nicely. So all these is possible of your TM. And these microscopes are quite rare in the world. So one of my friend, Aman Arora, he's a postdoc at the moment in USA. So he worked on a scanning electron microscope in Sweden, and it was only one of very few microscope were there in Europe also. So in India also, I'm not sure might have, but not so many. I think one or two, maybe in the IISc, Indian Institute of Sciences, or might be in some IITs. Uh, it's quite hard to maintain its maintenance. It's quite hard to maintain its level of thing. So we are done with our microscopy, or light microscope and electron microscope. Now how to prepare samples. So general for the light microscope, if you want to see something live, just view it, or have a vital stain you want to see it, then you view it. Otherwise, if you want to fix it, then fix it with the first you have to remove the water, dehydrate it, then infiltrate, and then mount on it, then make a stain of it, then you can view it afterwards. This is a very simple protocol to see your fixed specimens, and this whole process is known as histology. So there's a histology section in every hospital where and also in almost of the research labs also histology section is quite important, because whatever the study that you're doing on the cell level, you also have to see the things at the tissue level. So there your histology plays a big role in that case. So seeing something at the cell level, then bringing it to the tissue level, and watching it make a lot of sense then. To view organisms, tissues, or cells, so live mounts have to see the live things, yeah. So that's the main importance of live mounts. And fixed mounts is to keep the study of the tissue for a long time. And the chemicals that we used for them is a formalin, acetic acid, ethanol, methanol for fixations. And we can also use alcohol series to like from 30%, 40% up to the 100% alcohol. So here, if you want to see the section of your like an organ which is being in the gel or something, so you can check it with the help of vibrotome, which uses a vibrating knife to cut sections. So this is your specimen holder. You can put your holder here, and this is the knife holder, and it will just this is a magnifying glass. You can just keep it up, and this will be cutting it like this, tuck 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 tuck, and you can cut your specimens in different shades, and you can collect them here and start working with it. Then afterwards, so this is like how we in histology we do this. First step is as a formalin, then alcohol, then paraffin, or paraffin. With the help of paraffin we add our organ into that, then we do the sectioning with the help of microtome. Then afterwards we put the sections on the mounted on slides, stain to achieve the contrast of different components, and then we can check at the microscope. And we can also freeze them for quite number of time if you want to keep it, because they're in the paraffin wax, which could be let it be there like for a lot of time. So that was for the light microscope. But when you want to check for the electron microscope, scanning electron microscope, so this is the steps: fix, dehydrate, mount and stops, sputter, coat, observe. So these are the various things that you have to use while doing your SEM. So steps for them is as follows. First, you fixate it with the glutar, glutaraldehyde, glutaraldehyde, paraformaldehyde, and osmium tetroxide. Then further we dehydrate it with the specimens with the alcohol series up to 100% without water. Then to the organic solvents such as acetones and propylene oxide. Then specimens are processed in critical drying apparatus. Yeah. Then further specimens are mounted on the aluminium stubs like this. This is the aluminium stubs arranged in the holder. One of the stubs looks like this, and we stick them with sticky tape. Then Spooter coats specimen with the gold, palladium, rhodium in special chamber. Thick metal layer has been added. Then you can check them in your scan, absorb video out of it. So this is how your SCMs looks like, your scan electron microscope. Yeah. Then to check something for the TM, it's a little bit different. Here we have to infiltrate, we have to section and apply sections to grid, stain and observe. So in this case, we have a fixation specimens are fixed in again formaldehyde or glutaraldehyde, osmium tetroxide. Same way we have different series of alcohol series, then organic solvents, then they are infiltrated with epoxy or plastic resins. Then we have these molds where we can put your specimens from the molds. Then instrument ultra microtome is used to make your sections here. So instead of using microtome in the earlier case for the TM, we have an ultra microtome where you can have a specimen holder here and the knife cutter here. So this is how it works. So it will keep moving like this and this, yeah, and this will come in forward. It will also come back and forward. So when it is doing like this and this, so our sections will be cutted like this, and there's a water will be flowing here, and we can get our samples and bottom. So that's how this works. This is shown that how your different sections are being cutted like that. In the water they are coming up, ultra thin sections. And then you can use this ultra grid, metal grid sport to put your sample. Just wait a second. Yes, sir. Yes, sir. All good, sir. Okay, sir. It was what sir called to check all is fine. All is fine, case. Just give me a thumbs up. It's in CSI here. Okay, IGIP lab. Okay. Just thumbs up, students, if all is fine. Okay, good. So you put this like that, and then you can check under your sections in this TM microscope. So you have electron column here. Specimen insertion would be there. Additional images display could be here, and your fluorescent phosphorescent plate for viewing your image to be here. So that's it for today for this microscope chapter. Uh, I think we are good on time. Great, great. Thank you so much. So we are good on time today. We are done with the images. So we'll quit this. Um, Now what next? Now comes the tissue culture, which is quite important when you are done, because if you doesn't know how you can do the cell culture, you can't do any further techniques in this thing. So let's start with principle of cell culture. So animal cell culture. So here we'll be dealing with cell culture in vitro. So a brief history. So from it started from 1885. Rocks maintained embryonic chick cell alive in saline solution for a short length of time. Then in 1912, Alexis Carrel cultured the connective tissues and showed heart muscles over 2 to 3 months. Then in 1943, Arlil et al. Produced continuous red cell line. In 1962, Buonosi published methods for maintaining differentiated cells. And then in 70s comes the specific growth factors, media requirement for many cell types. In 79, Bortenstein and Sato defined serum-free medium for neural cells. And in 1980, tissue cultures become less experimental research and field for the accepted research tools. Then applications of cell culture techniques, so they could be helpful for study the cell behavior without variation that occur in animal. We can check like, what is happening in there, so we can check artificial organ development, in vitro propagation of virus, vaccine development, drug effect, recombinant DNA technologies. We can also characteristics of cells can be maintained over the several generations, leading to good reproducibility results. So whatever we are seeing in the cell culture, we can see at the long generation also. That is quite also helpful in that case. So finally, it avoids legal and moral and ethical problems of animal experimentations, because it says that in Germany, when I was doing research, that they were doing experiments on the monkeys, guinea pigs, mouse, all animals, that sheep, all animals you can think of, they were doing experiments on them. So there are a lot of ethical problems for that. So they say that if you can do things at animal cell culture, things will be might be happening at the organ level also, but it's not happening most of the time. But we can still save lot of money, lot of time, lot of things by doing experiments at the cellular level. So there are some limitations of cell culture. It has to develop standardized techniques in order to maintain healthy, reproducible cell for experiments. Take time to learn aseptic techniques. It takes some, yeah, 1 week at least to learn these techniques. Quantity of material is limited. Cost of media and maintenance of cell lines has to be taken care of. So there are many things has to be taken care while doing so. So some terminology in the cell culture, there is thing called organ culture. It's a three-dimensional structure of your tissues, disaggregated tissues, which you can see in the features of tissues in vivo. Then there's a thing called cell culture, single cell culture, no longer organized as a tissue, derived from the dispersed cell taken from the original tissue. And then there's a primary cell culture, which is just directly taken from your first tissue or any organ, isolated from there. And like in my case, what I show you, neonatal red cardiomyocyte, that's a primary cell culture where we have to how much is my battery? Just okay, battery is all good. Where I just showed you um, from directly from the neonatal rats, we had heart extracted, and then we just extract our cardiomyocytes. So these are primary cell cultures. So safety levels, so safety level has to be maintained. Substance that can cause cancer, carcinogenic, has to be away from there. It has to be, it could cause damage to the fetus or teratogen. Also mutation could happen at the genetic level, which could pass to further with the help with by mutagens. So like gentamicin, thapsigargin, just be aware of them. Even I use thapsigargin a lot during my PhD. I will show you these results also. Uh, Possible tetrogens, hygromycins, possible carcinogens, streptomycin mutagens. So these are the various, yeah, just be take care of it. So safety measures has to be taken care while in the cell culture, while working in the area. Do not bring your friends in with you. Like, hey, just join in. I am doing some voodoo science today, and I want to show you something. And do not eat or drink or smoke in these areas. Do not use mobile phones. Wear lab coat always. Do wear disposable gloves also. And throw them at the correct way. In our case, we were working in the hospital, so we have to wear this PP kits also, because we were working with the human cell culture. So we have to maintain really high level of safety measures in our labs. So my lab was actually when I was working. It's in the hospital called Universität Klinikum, University of Klinikum. Klinikum means hospital. So it was a hospital, a University of Hospital in there. It's in the Göttingen. It's in a city called Göttingen, where most of the Nobel laureates are there, and yeah. So it was a research lab also, and also a teaching for the students. It's like our aims actually, like we have aims in Delhi. Uh, so yeah, basic equipments have to be maintained, like horizontal laminar airflow where you can work with the things. Sorry guys, there's a lot of text there, but I'm pretty sure I will show you some videos and some more better presentations where you can see some direct explanations like how things are. Uh, Then filtered air should be entered into the cabinets, tracked into the front and directly to the operator. Cabinet has to be sterile. Your centrifuge in cell culture area, human-derived cells must be centrifuged in the sealed rotors. All these things has to be maintained. If sometimes contamination could happen because of temperature, pH, or nutrient is not good, or its division of the cells becomes too much, or there's a mycoplasma infection, so you have to clean everything from the start, and your all experiments will be maintained for that also. So development of antibiotic resistance mechanisms is also required while doing so. So how you can isolate cell lines for your in vitro culture? You just resected a tissue, then get your cell out of it in vitro. Then you make a primary culture. Then you have a secondary culture. Then you make a cell line. Then from the cell line you can do a single cell isolation, immortalization, and successive subculture. Then it could also happen. The senescence could happen. Transform cell lines could happen, and then clonal cell lines. And then from the immortalization, immortalized cell lines could make. This is how your isolation of cell lines are happening in vitro culture. Now, types of cell culture in vitro we have discussed before. Primary cell culture is the first one we're gonna discuss. So these are directly derived from the animal tissue, embryo or adult or normal or neoplastic, any of them. They retain differentiated phenotypes, mainly encourage dependent, and exhibit contact inhibition thing in it. Then there's secondary culture, it's derived from the primary cell culture. Like if I take my those cardiomyocytes that I've showed you, and from them if I start to make another culture, that would be a secondary cell culture. It mainly reason for cloning or selection of the things. It has finite lifespan in vitro. It's not has infinite life of span. Of course, after a few days they will be destroyed, and it exhibit contact inhibition with others. Then comes your continuous culture. In a continuous cultures are derived from your primary and secondary culture itself. They are immortalized. They can spontaneously divide as much as they can. So by transformation vectors like viruses or plasmids, serially propagated in culture showing an increased growth rate, homogeneous cell population, loss of anchorage dependency, infinite lifespan in vitro. So these are the, and they are not genetically unstable actually. So this is, you can see fibroblast on this side, and this is epithelial cells, and these are neuronal cells, these are endothelial cells. This is how various cells, every cells has different shapes, different morphology, different way of movement. On that note, I can show you very, uh, what is Binod? I don't know. Uh, so guys, so as I showed you those microscope things, so here what you can see is the video sequence of on the left side is the various phallopodia. You can see how the cell is moving from one place to another. Just check these beautiful videos. So this is how from the back how it's getting pushed and moving forward. How from the back it's getting pushed, moving forward. Just check this. So this is how the movement is happening on this side. On this side your cell are moving forward like this. This is called cell motility. So there is a big science behind this. Is interested. There's a lot of groups are working in the same manner on the cell motility. Okay. This is done with the help of DAPI and fluorescent dye. So you can see the movement of cell. So how you can see beautifully they are moving in front. So from the back there's a push like this. Then again in front push. So nice figures, very nice figures. I think that's enough for us. Pushing and pulling. This is what pushing and pulling is happening. This is a group call from the Iowa Salex something. So we have discussed this already. There's a primary tissue culture. There's a continuous which can have finite and indefinite. So main type of we already discussed primary and continuous cultures. So this is from a morura you can make a blastocysts, and then from blastocyst you can make inner cell mass, and then embryonic stem cell culture, then further muscle cells, blood cells, neurons, intestine cells, pancreatic islets, liver cells. So you can make a lot of from your embryonic stem cell culture these different kind of your tissue level cell culture. Then from the stem cells, they have ability to self-replicate and differentiate to multitude of different cell types needed within the body. They can make many different types of, and they can happen by them. So you can see they can differentiate into mesoderm, endoderm, and ectoderm. If the mesoderm could form into red blood cells, skeletal muscle cells, cardiac muscle cells, whereas endoderms could form pancreatic, lung, thyroid, and ectoderm could make pigment, neurons, skin cells. Further, self-renewal is pluripotent stem cells could happen on the top. Yeah, this is the same diagram further. So you can check. This cycle of our cell cycle. G1 phase. The spindle assembly checkpoint. G2 phase. DNA synthesis phase. You can check also in this case. Degree of transformation we can check with the degree of epigenetic changes. So these were the various applications you can do with the help of culture. Yeah. So cell culture environment, how it should be, it should have nutrients, environment, carbon dioxide, temperature 37 degrees Celsius, humidity has to be maintained in the incubators, sterile, it has to be aseptic techniques, we have to use antibiotics, a lot of anti mycotics has to be used while doing so. Yeah. And then cell culture environment, how it has to be done, it has to be done in the basal media it could happen, or what are the it has maintained pH and osmolarity. All the osmolarity and pH is maintained, provides nutrients and energy source. Components of basal medias are inorganic salts, pH indicator like phenol red, buffers, glucose. So these are the main components. Whenever you buy media for your cell culture, just read what are the things it has. It might have a regulate membrane potential, sodium, potassium, calcium, which will maintain your membrane potential of it. Inorganic salts will maintain the osmolarity of your cells, and it also helps to attach ions and enzyme cofactors to it. So there's to check, because the pH is of your cell culture is 7.4. So whenever the pH goes up or high, acidic or basic, you can check it by the change in color also. Then there's buffers called bicarbonate and HEPAs are added, which are requires carbon dioxide atmosphere. So these are added because for the carbon dioxide. This is quite important. I have to admit, important. Slide from the perspective of what are the basic aspects of cell culture, what are the basic medias that are added, and what is the reason why we are using them. And then HEPA strong chemical buffer range of 7.2 to 7.6, which does not require carbon dioxide, and then it's a glucose for the energy source in it. Also, further, we need carbohydrates, vitamins, trace elements, keto acids. So these are our requirement for our basal media. And then supplements are added sometimes: L-glutamine, non-essential amino acids, growth factors and hormones, antibiotics and antimycotics like penicillin, streptomycins. So just remember all these. These are very important because when we were creating media, so for us it was a uh, day-to-day work. So we were adding them already in our media, and then we start to do our work culture. So if one of them is not there, if your growth hormone is not there, if you're not adding any antibiotics to your cell culture, so some you can have some problem later on. So all these supplements, all these media components, they are quite important in your cell culture. So these three slides are very very important, very very important if you are gonna start your cell culture. Because whenever, if in your life in future you're gonna start your cell culture, if you have these slides, you will know that okay, Simran sir told us about these components and why we are using them. You will know this, and you can tell to your PI also, your principal investigator, that we are using them for following reasons. Then cell culture environment, also other in vitro culture, fetal calf bovine serum are also used in that case. So which are growth factors and hormones which helps in the cell attachments. Yeah, it's being used quite lot, quite a number of time before. It has some disadvantages as follows. Further on, we have to heat inactivate these FCS and FCS. Why heat inactivate? For 56 degrees Celsius for 30 minutes in water bath, to destroy the complements and the immunoglobulins, to destroy its all the immunoglobulins, and to destroy its all viruses, which are by gamma irradiated serum. Don't do it overdo your heating because this could damage the growth factors, hormones, and vitamins, which could affect cell growth. So this is, you can see, contaminant level 2 cell culture laboratory. This is the airflow lamina, and this is your typical cell culture looks like the flask, and this is Mr. Frosty. This is used to froze your cells at minus, I don't know, minus 90, minus 70 degrees Celsius or so. So in the cell culture, how do we culture our cells at the laboratory? First of all, we revive frozen cells population, isolate from the tissue. So we have revived our cells from the Mr. Frosty and maintain in the culture like this in the typical flask, and with the help of aseptic techniques. Then we do the passaging subculture, and then we count the cells, and then after counting the cells, we again cryopreserve it at minus 70 degrees Celsius. So why passaging cells? It's maintain the cell culture. We do not have overgrow in our cell cycle. So you can see this side on the left is 70 to 80% confluent. On the other side, it is 100% confluent. So how we do it? We remove the spent media, whatever the cell culture we have, we remove that media and wash it with PBS. Then we incubate with the trypsin EDTA and resuspend in serum, and then transfer to culture flask. So on passaging, please students, all is good. Just give me a thumbs up. We are going good today. No lagging. Voice is fine. I'm slow. So I have 5 minutes video for passaging of cell culture. Let's start this.
The standard pattern. A leg after seeding is followed by a period of exponential growth called the log phase. Cells should be passaged when they cover the plate, or the cell density exceeds the capacity of the medium. Maintaining log phase growth will maximize the number of healthy cells for your experiment. Before retrieving your flasks from the incubator, sterilize the hood and have all required supplies at hand. Examine your culture carefully for signs of contamination or deterioration. Handle the cells with care during transport. For adherent cells, remove the spent medium from the flask using a sterile pipette. Rinse the cells with a balanced salt solution, such as DPPS. Make sure to use a salt solution without calcium and magnesium, as these may inhibit your cell dissociation reagent. After rinsing the cells, remove the salt solution. Each time you aspirate liquid off the cells, place the next solution on quickly. Add your cell dissociation reagent to remove cells from the plate. Use just enough solution to cover the cell sheet. Consider using a gentle dissociation reagent, such as Triple Express, to avoid damaging your cells during dissociation. Make sure that the solution completely covers the cells. You may want to tap gently on the plate to help the cells detach. Use a microscope to confirm the cells have released from the flask. They will start to appear round as they release from the substrate and will move or slide when the flask is tilted. Complete dissociation is necessary. Do not leave the dissociation reagent on too long, especially if you're using a reagent other than triple. You may choose to manually break up lingering clumps by repeatedly pipetting warmed medium over them. A single cell suspension is important to achieve an accurate cell count. If you are using trypsin, the collection medium will need serum, or you'll need to use a trypsin inhibitor to inactivate the trypsin. If you're using triple, inactivation is achieved by dilution alone. No serum is needed. Transfer the cells to a conical tube and centrifuge to remove any residual dissociation reagent. The speed and time of centrifugation will vary based on your cell type. After centrifugation, you should have a well-formed pellet. Remove the medium from the centrifuge tube with a pipette and discard the medium into a waste container. Try not to disturb the pellet. Resuspend the pellet with warm, complete growth medium. Gentle pipetting will disperse the cells to ensure a homogeneous solution of single cells. Remove a small sample for cell counting. Trypan blue is used when counting cells to indicate the ratio of live to dead cells. The stain turns dead cells blue, but healthy cells with an intact membrane will remain white or colorless. Using the microscope, count the total number of cells and the number of dead or blue cells. Based on your cell count, determine how much additional fresh medium to add for optimal seeding density. Add the required medium, mix the cells gently, and pipette the solution into fresh flasks. Cap the vented caps on your flask tightly. If the caps are not vented, cap loosely. This keeps contamination out while allowing adequate gas exchange. Use a north-south, east-west motion to evenly distribute the cells and transport the flasks to the incubator. When passaging suspension cells, you'll begin by removing a small sample from the cell culture flask for counting. You'll follow the same counting procedure for adherent cells using Trypan Blue and either a hemocytometer or the Countess automated cell counter. Based on your cell count, add additional fresh medium to the flasks. Stay within the minimum and maximum volumes. This is important to maintain optimal air exchange and shaking flow. You may need to split the culture into multiple flasks. Cap the flasks appropriately, depending upon whether they are vented or not, and return them to the incubator. Every 3 weeks, gently spin the cells, remove the medium, and replace with completely new medium. This eliminates cell debris and metabolic waste.
So that was exciting. That was quite good summary what you have studied uh, so far, and quite good one. So whatever I have taught you today, it is being shown here very nicely, everything in good summary. So I will continue still. So passaging how you can cryopreserve your cells. You can do your with the passage cells with the help of resuspending cells in the serums, then centrifuging the aspirant supernatant, and then resuspending the cells in the 10% DMS on FCS and transferring to the cryo vials and transferring to the nitrogen tanks. So why we do the cryopreservation is just to have reduced risk of microbial contamination. So we keep our minus 80 degrees our cells so that we can use them for the future long and there's no contamination and we can have better things. So this I already told you, manual cell hematocytometers, where you put your samples with a tryphon blue and the cells that which are dead will be on the round, not the red, the one, the cells which took the tryphon blue would be seen in the here in the scare form. Then average of cell number multiplied by the dilution factor of cell suspension multiplied by 10,000. This will be your cell count. And there are also automated cell count. You just put your cell into the chamber and then let you know the automatic of it. So this is the ideal, this we all know, ideal growth curve of the cell culture. First is the lag phase, then is the exponential phase, then is the stable plateau. um, there is a cells per ml, then the days from the subculture. So we can see that how first initially it will be lag phase, then suddenly they will grow, then at the moment they will maintain a plateau. Sometimes it's unstable plateau because of rapid decline, some mycoplasma infection or so, something like that. So contamination we have discussed, mycoplasma contamination, bacterial, yeast, or any contamination could happen at any level. So yeah, this we have discussed with the embryonic stem cells, how you can create a lot of other different parts. Yeah, we finite cell line, continuous cell lines, their property, how things are, looks like. So these are the common cell lines: human cell lines, MCF7 breast cancer cell line, HLS leukemia cell line, HEC cells, this is human embryonic kidney cells. These are the most likely HeLa cells. So these are the most common cell lines that are used in the lab. If somebody is working, I don't know, in the lab, you might be using these cell lines somewhere or somewhere not. So this is how your organ culture looks like. So you can see artificial organs are put it up in these conical, not conical, cylindrical flask. Your This is your lung, I guess. This is your heart. This I'm not sure what is it. So these they are made these artificial organs actually to maintain them. So thank you very much for the time. Uh, we are on good time today. Uh, Human intest. Oh, just don't spam, bache. So, can you please give insights on the fermentation on human intestinal cell culture? So, yeah, I mean, for you can do that also in that case, but as we have discussed about various cell culture, cell lines, various aspects of things. So you can do also this fermentation, like in the fermenter, you can make that thing also. So let's continue with the last slide of today. So we go slowly now, not so fast. Uh, so why cell culture? Cell culture could help you to the gene therapy, embryo culture, primary human and animal cell culture, stem and cancer cells, make proteins and also antibody productions and monoclonal ones. So we have discussed why we do it with so much of advantages we had, and it's also in the tissue bench top should be kept clear and clean. This we have discussed already. Um, no need to discuss it again. So primary culture is a fresh isolated, where the cell line is a finite cell line which dies after several subculture, whereas continuous cell line is transformed, or the immortal ones. So passaging, as we had discussed already, how the passaging is done. Cell dissociate from the flask using enzymes, split into two flasks, and we can go it for further things. So your cell culture is contact inhibited. Your cells are at the bottom, but with the time, if you don't change your passaging, they will keep uprising up. So this will leads to the very mess cell culture. So cell can be initiated from tissue or organ. We have discussed that. Cell single cell suspensions. Choice to be made whether you want to change the media, culture conditions, selection procedure. All these things matters. So these are the various considerations you have to take care while doing so. Mm, this is not so important for you all. Yeah, this I want to show you actually. You can see this is 8 culture dish and this is 96 well plate culture dish. So here you can grow in this 96 well plates. Uh, the reason to show you this presentation is to bring you to the topic of my master thesis that I did at Max Planck Institute in Germany, with the in the under the guidance of Professor Offermans and Dr. Boris Strilich. So there we were working with the metastasis, cancer metastasis, and we did siRNA transfection on Huvik cells, and then check after the transfection on the Huvik cells on 96 well plate, what happened to the cancer cells, whether they can transfer to the human embryonic Huvik cells or not. So to do so, from this presentation, I will switch to the my master thesis presentation in order to let you understand. This is a high throughput screening. Actually, this technique is known as high throughput screening to check. This was done in 2012 under these professors. So my first referee was Dr. Katja Becker, then second referee was Dr. Stefan Ralphs. My supervisor was Dr. Boris Strilich, and the name of the institute was Max Planck Institute. The main reason that I'm showing you is this to have the understanding of this all cell culture that we were studying, how they could be applied in your field, how they could be used in day-to-day research actually. So a little introduction. What is metastasis is? Metastasis is something like, you can see there's a primary tumor. From this primary tumor, you have a spreading of tumor cells. So the invasion will happen, and they will come to the to your organ cells. They will just go transmigrate to your Huvik cells. So this is called transendothelial migration. Intravision is happening. Then they will come inside. They will adhere with the help of adhesion. So inside your blood or lymph vessel is moving. Then from there, from the transendothelial migration will again happen, and here it's called as extravasation. Then they will invade to the secondary site, and then it's become a secondary tumor. So it's from primary tumor. This is how a secondary tumor is made, and this is whole process is called as metastasis. This whole process is called as metastasis. And then there are GTPases, your um guanine tyrosine phosphatases. Yeah, so they are responsible for things. So I will not go in detail with them. So they are mainly these different kind of rho GTPases, like this rho GAP59. So we had these siRNA libraries. So we have these 59 siRNAs, we have 65 siRNAs, 22 siRNAs, 20, 17, 35. So we have this much of 218 genes in total while doing this siRNA transfection. So here you can see seeding of Huvix is happening. We added our Huvix first of all on those 96 well plate chamber. Then there's the EGM medium and there's upper chamber called free. Then knockdown is done with the help of siRNA, and afterwards, after the knockdown, transmigration assay is happening. So this is for 5 hours. We kept it them there. So you can see some cancer cells are being transformed to the bottom after the transfection, and the phenotype of our cells are changed. And then at the bottom you can see some tumor cells being there. Yeah, some automated tumor cells are being there. So you can we can check them via microscope. So how much of transmigrated tumor cells are being done with the after the transfection. So any questions so far? Yeah, all these presentations will be done. Don't worry. Transfection, bache, it's something to knock down the cells. Uh, to knock down your cells, to remove the effect of one gene from your cell line, we do the transfection. Yeah, all good so far. Okay, just give your thumbs up. Okay, I continue then. So then further, oh, not this one. So this is your Huvix cell X looks like in that 96 well plate. So there are 96 wells. So we added these Huvix cells on top. What we saw that that Huvix cells at the outside layer, they were not so nicely present. They were like, yeah, badly constructed, and a not so good way was happening. So we remove these outside outliers and we just try to keep the cells inside these cell lines. So we optimize this, and afterwards we just kept our cells to be inside. Then we had these siRNA plates. We have these controls, CDH5 killer cell line. So these are our controls. All these controls they outside, and we also have calcine there to check whether our cells are present or not. We're adding the green fluorescent light dye to it, and these are various siRNAs we have added to it. So this is what the monolayer of our Huvix cells looks like. That okay, our cells looks like the same like in our body. Uh, they maintain a monolayer. That is confirmation. These are the controls. With the CDHF5, we see some destruction of the cells, but the natural killer cells totally destroyed. So we kept them these always on the side to check that whether of the Huvix cells are being destroyed in this concentrated manner. Like it's okay, it's more destroyed, then it's more destroyed. So then we calculated the after cutting them how much tumor cells has been transmigrated. So we calculated them. So the more we kill the Huvix cells, the more the cells are transmigrated. The more we kill the cells, the more cells our cancerous cells are transmigrated. So this was a quite a nice control when we are doing our experiments. So controls are quite important when you're doing experiments. Your negative control, your positive controls, keep them in remember. If you can maintain a good control when you're doing experiments, you will be sure. It's also in the daily life when you are like fixing any house things, just make controls out of it so that there will be no problem at the end of your experiments. So this was something to do with the software. This is the number of transmigrated tumor cells out of it. Then we analyzed them. This is Huvik monolayer. So we found around uh, 25 express genes to be dealing with the high transmigration of tumor cells, and these were the non-expressed ones which didn't express. We did the siRNA, so this I cannot share further. So this it looks like final hit distribution. So there were some lower outliers, some higher outliers. So these might be the one which are the genes which were leading to the transmigration of cancerous cells to be highest. So if we can delete these genes in the human body, we can save the patients. So that's it. Thank you for your attention. So that's the main reason. That's why I bring you to 96 well plate cell culture. So when I did that cell culture, uh, I want to bring you to this one video that how this 96 well plate work. So this is automatic robotic. So that's it. You can see how these robots were doing your whole experiments. I did that actually manually when I was doing these on hand, but here robots were doing everything with the help of this is quite old video actually. This is happening in USA. USA, the research is happening at very large, very like high tech. It's even lacking the Europeans behind. Germans are still a bit behind than the States people. I have to recommend. And India is still fighting, and it's still somewhere it has name, but not as the Europeans or States people have. So this we have to admit. So this was about 96 well plate culture. Uh, So factors affecting cell behavior in the environment, cell-cell interaction, proteins, cytokines, hormones, all these have these differences. So cell culture surface, most adherent cell cultures dash to the proliferate. So change in the charge of the surface, polyl lysines, all these are happening. Media formulations, DMEM, like we discussed in the previous FBS media, basal media. So these are also called DMM media. So this is the various components of it. So these are various amino acids are being added in that medium: glycine, alanine, glutamine, arginine, hypochlorite, L-cysteine. Vitamins are added. All inorganic salts are added. Other components are added. So these are good to know, good to remember, because they are quite important ones. Then media formulations are happening: inorganic ions, trace elements, amino acids, vitamins, energy sources. So we have discussed them already. So serum provided, we have already in that part discussed. The phase has to be maintained with oxygen and carbon dioxide. How much we have to give? These are the carbon dioxide incubators with the controlled carbon dioxide and humidified 37 degrees Celsius in it.